Related to the third goal of Sustainable Development Goals 2030, especially to eliminate of neglected tropical diseases such as dengue hemorrhagic fever, based on records from the World Health Organization, an estimated 500,000 DHF patients need hospital treatment each year. This disease is included in the 10 causes of care in hospitals and other health facilities and deaths in children in at least 8 tropical countries in Asia. Various efforts have been made in anticipating an outbreak, start from using the anti-mosquito spray to lotion. However, research showed that anti-mosquito drugs are not entirely beneficial. The use of them can leave the residual residue, pollute the environment, and can cause the resistance. Residual residue can cause health problems such as irritation, muscle spasms, and disrupt the respiratory system, and even triggers the occurrence of asthma and cancer. Therefore, concrete efforts that are safe and effective for humans in the environment are needed. One of them is by applying an idea from me, Refa Rahmaddiansyah from Indonesia, entitled Bakulis Air Spray Solution from Jeruk Limau Peel Waste and Lemongrass as Preventive Solution to Dengue Hemorrhagic Fever. Hemorrhagic fever can make a patient's body's temperature very high, usually accompanied by headaches, joint pain and muscles and bones, and pain in the back of the eye. This disease is caused by dengue virus, which spreads through the bite of the Aedes aegypti mosquito. This mosquito breeds along in densely populated area with humid and warm climates. The purpose of this study was to determine the effectiveness of Bakulis as natural herbicide and anticipating the HF as an effort to achieve the SDGs for all related sectors. We estimate this innovation can provide benefits, especially in efforts to overcome the tropical infection and reducing mortality. This research is purely experimental research with post-test only control group designed by making airspray solution products from jeruk limau peel waste and lemongrass, which are analyzed descriptively and analytically. Experiments carried out with randomized control design, 3-time repetition to analyze the effectiveness of this product. This study used two factors: positive control using infusion products while the negative control using aqueous in the control container. Besides, we use the hedonic or genoleptic test for measuring public likeness of this product. This research was started from collecting the ingredients and tools, infudation method of extracting process until it's done as ready-made product of Bakulis. And here are the prevalence of the respondents that based on hedonic organoleptic test, all the people like this product. For testing the toxicity, there were no reports of toxicity from responders, either from the allergic reactions or other acute effects. So, what contains of jeruk limau peel waste and lemongrass? Previous research stated the results of the phytochemical test of lime samples from the peel extract all gave positive test results for the content of alkaloid, tannins, saponins, and flavonoid. As for lemongrass extracts, it gave positive test result for the content of saponins, tannins, alkaloids, quinones, and steroids. This contents is due to kill the Aedes aegypti test larvae by inhibiting the action of the acetylcholinesterase enzyme on nerve impulses and reducing their ability to digest food. This bioactive compound is the cause of larval death because it's unable to detoxify the toxic compounds that enter the larva's body. The mechanism of action of larvicides in killing larvae in three ways, such as contact poisoning through the body wall of the larvae through the contact with the skin, penetrating the cuticle, trachea, ancillary glands, or the other organs related to the cuticle. The body wall of insects is the part of the insect's body that can absorb the large amount of toxic substance through the stomach poisoning by moth and as a respiratory poison of fumigen. So we can conclude that this solution was proven to be effective as a natural herbicide against the manufacture of dengue hemorrhagic fever and the Aedes aegypti mosquito. As for the result of the favorite level test, the satisfactory results were obtained where all respondents liked the product. Has a potential to produce massively. And it is suggested for the further research to determine the optimal dose by increasing the number of control groups and various concentration. Here are the references that I used. So, ladies and gentlemen, that's all my idea and innovation. Hope you can interest and it can be applied to solve the problem. Save your health and explore the nature. Stay safe and thank you very much for this chance.